Now, despite Dolly's unusually short telomeres at birth, she didn't seem to have any other major health complications. She was, in fact, the model of a healthy clone. However, as years progressed, it was discovered that Dolly was diagnosed with arthritis at an early age. This seemed unusual and made scientists worry that perhaps the unusual age of her cells also made her more predisposed to health issues found only in older sheep. But this was unlikely a result of the telomere shortening, and much more likely a result of the fact that Dolly herself was kept indoors almost her entire life. That was because she was protected from animal rights activists that wanted to free her and take her out of captivity and out of scientific research. And so the researchers kept Dolly indoors all the time and closely watched. As a result, she didn't get enough exercise in her life, as much as a typical sheep would, and ended up developing arthritis. Dolly also eventually contracted a lung disease and died. Again, lung diseases are common in captive sheep, so this was probably not a problem from cloning, but rather an issue that arose out of her lifestyle. Subsequent to Dolly's cloning, other animals have been cloned as well. And after cloning these other organisms, scientists tried to understand if there were any other possible health complications. The most commonly acknowledged risk of making clones is the failure of making a healthy clone. It's in fact quite difficult to make healthy organisms when cloning an animal. Roughly 90% or more attempts at making clones is unsuccessful, meaning a vast majority of the attempts result in either no live births or defective animals at birth. Cloned mice are also generally less healthy than non-clones. This is not really a change in genes or the cloning process, but in how the genes are used by the animal itself. This is what's referred to as gene expression. The use of genes, or gene expression changes, are known to occur as genes pass through egg and sperm. Gene expression differs between whether or not you inherited the gene from a mother or a father. This is what's known as imprinting. If certain genes are passed through the father's lineage, some of those genes are used more often, as opposed to other genes, if they are passed through the mother's lineage, they are used less often. In fact, functioning adult cells require that there are two copies, one which is imprinted from the mom, and that's of course inherited from the DNA from the egg, and the other is imprinted from the dad, and that's from the DNA inherited from the sperm. Because all of the DNA would have been inherited from the single sex from which it's cloned, in the case of Dolly, it was her mother, all of the DNA would have had female imprints on them. And because you don't have equal imprinting, one from mom and one from dad, this could create difficulties throughout lifetime. In fact, if you've heard of recent discoveries of being able to produce offspring from two organisms of the same sex, then you'll understand what a challenge that would have been for those scientists, because both of those DNA copies were inherited with female imprinting. Scientists have yet to be able to generate cells with imprinting of males exclusively. Now, what does this imprinting have to do with a healthy life? Well, some of the narrow features associated with organisms that have been cloned have identified that there could be some immune system disorders and also perhaps a shorter lifespan. This has only really been shown to be mildly conclusive in mice, though, and any clones of farm animals, sheep, horses, cows, and others, have found no main or major health issues. As a result, horses that have been cloned are now allowed to be used in competitive races. Cows that have been cloned are allowed to be used in our food supply. And the FDA and USDA has all certified that cloning farm animals is okay and healthy for the organism. There are, in fact, lots of reasons organisms have been cloned. For research, there are some organisms like mice, rats, rabbits, and even monkeys that have been cloned to better understand the cloning process. Monkeys in particular were cloned to see if it would be possible to clone a human. Human infants have not been cloned yet, but human cell lines have. None of those cells have been allowed to progress to a human infant, child, or adult, however. Other researchers are investigating the use of clones for producing pharmaceuticals or drugs, and that includes cloning pigs and goats. Pigs and goats can produce pharmaceuticals or drugs in their milk or other bodily tissues, and then those tissues or milk can be harvested and the drug purified for human health. Cloning, of course, has also been used as an attempt to recover endangered animals. Included in this is a gaur, which is a type of ox, a mouflon, a wild sheep, a Pyrenean ibex, which you can see here. This one was brought back from extinction for only 7 minutes, but then died after 7 minutes as a result of complications from the cloning process. An endangered ferret was cloned this way, a wolf, and also a water buffalo. Now for pet owners were also involved in the cloning process. Cats, dogs, and horses have been cloned. And as I mentioned, horses have also been cloned for races, but also primarily sperm production. So if you have an award-winning racehorse, some of the most money you can make out of that award-winning racehorse is the sperm that you can sell. This is also true of cows and mules. Prized cows in our food supply and prized horses and mules can have their sperm donated, often for $1,000 per ejaculation, and a lot of money can be made. And since it's a clone, and you're not asking it to race, you're just selling sperm, you can make a lot of money off of this organism. In fact, this has been done for the past decade or more. To the point now where, as I mentioned, the FDA and USDA said it'd be okay to sell meat from cloned animals, and also the racing agencies in the United States have certified it would be okay to race a cloned horse. Now, there are some ethics of cloning as well. The pressure to create a cloned human is really high, creating an environment that has high reward. Hwang-Woo Suk, a scientist in South Korea, was the first individual who cloned a dog. The dog that he cloned was named Snuppy. Snuppy was cloned in a process similar to Dolly the sheep, where you had the Afghan wolfhound donating a cell, and that cell was then mixed with an enucleated egg cell from essentially a mutt. And these cells were fused together, placed into a Labrador surrogate mother, and then that Labrador gave birth to a cloned puppy. This is also how people clone their pets today for about $20,000 to $30,000 per cloned animal. Now, he gained a lot of notoriety for cloning the first puppy, and a lot of companies ended up using his technology and research to be able to advertise and sell this service of cloning your pets. Subsequent to that, he was put under further pressure to clone human embryonic stem cells. And so in 2004 and 2005, he had claimed to clone human embryonic stem cells. Again, not cloning a human, but cloning a cell line. The data was also published in peer-reviewed scientific journals. About 6 months after that paper, the second paper was published, news reports hit claiming that women were paid for their eggs, something not considered ethically sound in the scientific field. And also, women in his research group were part of the egg donor group, and some of them claimed to have been pressured into donating their eggs. Egg donation is a very dangerous process and can, in some cases, result in sterility of the donor. And so donating eggs is no small feat. While this is not illegal, this is considered unethical, and it resulted in further probing of his research claims. Within 1 month, the university declared the human stem cell cloning data was fabricated. And in that time, in that 1 month time, Huang Wusek eventually was fired from his position and put under trial for stealing from the university as a result of receiving grant funds from his fabricated research. He was sentenced to 2 years in prison in 2009 and found guilty of embezzlement and bioethics violations. This process, again, exemplifies how important the peer review process is in scientific literature these days, and how diligent and vigilant the scientific community is in checking and rechecking data and experimental procedures. The fact that this very famous scientist had discovered to have fabricated data in his human stem cell lines, and all of that was discovered within 1 month, was laudable. Importantly, though, his research in the cloning of animals was not falsified. And as I mentioned, that research is still being used today to clone people's pets. So that's it for our animal cloning lecture. I hope you learned a lot. Would you consider cloning your pet? Bye.